All right, class, welcome. Today, we're going to dive into one of the most elegant and honestly, one of the most powerful tools in all of modern neurobiology. It's a genetic switch that lets us control the very circuits of the brain. But, to really get why this thing is so brilliant, we got to start with the huge problem it was designed to solve. I mean, think about it. How do you even begin to map the brain's impossibly complex circuits?
You might look at an organism like a fruit fly and think, how complicated can it be? Well, let me tell you, its nervous system is an overwhelming tangle. It's this incredibly dense, intricate web where thousands of neurons are all firing together to control everything, from flying around to finding food to really complex stuff like courtship.
So for neuroscientists, for the longest time, the big problem was one of specificity. How can you figure out what just one gene does in just one type of neuron without messing up the thousands of other cells right next to it? It was kind of like trying to fix the wiring in one single house by shutting off the power of the entire country. The tools were just way too blunt.
So where did the solution come from? You might think some complex mammal, right? Nope. The answer, brilliantly, came from a really unlikely place. A protein found in common baker's yeast. And it works like a two-part genetic light switch, built for incredible precision.
Okay, so let's break this down. The first piece of the puzzle is the switch itself, a protein called GAL4. Now, GAL4 is a transcription factor, but don't get bogged down by the term. All you really need to remember is that its job is to bind to DNA and turn other genes on. Think of it like a master key, a key that's designed to fit one very specific lock. And of course, every key needs a lock. That's our second piece, the socket, which we call the upstream activating sequence, or UAS. Now here's the really crucial part. This DNA sequence doesn't naturally exist in a fly, at all. So nothing else in the fly's body can turn it on. Only our GAL4 key can. That absolute specificity, that's the secret sauce to the whole system. Okay, so this is where the genius of the system really shines through. The key and the lock, they aren't put into the same animal to start with. Instead, they're engineered into two completely different, separate lines of flies. First up, scientists create what's called a driver line. They put the gene for that GAL4 key into the fly, but they put it under the control of a very specific promoter. A promoter is just a piece of DNA that acts like a cellular address label. It makes sure a gene is turned on only in specific cells. So if they use a promoter that's only active in courtship neurons, well then only those cells will ever make the GAL4 key. Then, in a completely separate responder line, they take whatever gene they're interested in studying, let's say a gene that makes a green fluorescent protein that glows, and they stick it right next to that UAS lock. Now, in these flies, that glowing gene is in every single cell, but it's totally silent, because there's no GAL4 key anywhere to turn it on.
So, on their own, these flies are, well, pretty boring. Nothing special happens. The driver has a key, but no lock to open. The responder has a lock, but no key. But then, you cross them.
And in their offspring, bam! That's where the magic happens. For the very first time, the key and the lock are in the same animal. The GAL4 protein, which is only being made in those specific courtship neurons, finds that UAS sequence and flips the switch on our glowing green gene, but only in those cells.
What's so incredible here is that this simple genetic cross isn't just one neat trick. It's like opening up a massive toolbox that lets neuroscientists see and even control the brain with a precision that was just unthinkable before. And just look at this. This is what it lets us do. On the left, that's the big messy tangle we started with, right? But on the right, wow. Using a driver for neurons that express something called neuropeptide F and a UAS GFP reporter, a single circuit is brilliantly lit up. We can trace its exact path through the brain, totally isolated from all that background noise. But just seeing them is only the beginning. We can get really creative. Instead of a fluorescent protein, we can stick a gene next to the UAS that activates neurons, letting us artificially trigger a behavior. Or we could use a gene that silences neurons to see what function is lost. We can even use cell death genes, like one called Reaper, to completely remove a set of neurons from the circuit. It's an incredible toolkit.
And that right there, that is the crucial breakthrough. We can finally move from just observing things to actually doing experiments. We can ask incredibly precise questions. If we activate these cells, does the fly start a courtship dance? If we silence those same cells, does the behavior disappear? For the first time, we could directly and definitively link a piece of the brain to a specific function.
So, let's put this all in perspective. How big of a deal was this, really? Was it just another tool, or was it a revolution?
Well, it's hard to overstate it. When Andrea Brand and Norbert Perimon published this back in 1993, it just changed everything. They were pretty modest in their paper, but the rest of the science community saw it for what it was. It was immediately hailed as, and I'm quoting here, one of the most important advances of Drosophila genetics, a total game changer.
What it gave us was a recipe, a clear, logical, repeatable playbook for discovery. Step 1, isolate a circuit with a specific GAL4 driver. Step 2, cross it to a UAS line to do what you want. See it, turn it on, turn it off. Step 3, measure what happens to the fly's behavior. And step 4, conclude. It turned the overwhelming complexity of the brain into a series of puzzles we could actually solve.
So in the end, the GAL4/UAS system is basically a master key to the brain. It gave us a way to finally start unlocking the genetic and cellular basis of everything from learning and memory to behavior, one single circuit at a time. The only question left for us is, which of its countless doors should we open next?